Okay. So in the last class, we had discussed about fermentation of dough, right? Janvi had done it. Okay, what happened, Sakshi? Your dough did not rise.
Yeah, my dough hadn't rise, but then I tried again,
Okay.
and I still have to see whether it will rise or not.
Okay. So what you'll have to do first, again, I'm explaining. Okay, what you'll have to do is you'll have to take lukewarm water. All right. You'll have to take lukewarm water, add sugar to it, stir it well, dissolve the sugar completely. Okay, and then add yeast to it. After about 10 minutes, okay, leave the yeast to activate inside the sugar solution, and after that you can mix your dough with that water, with that sugar solution. Okay, and then you can keep your dough for about 2 to 3 hours and observe what happens. Okay, after 2 to 3 hours, what you all will see that the dough has risen. Okay, you all will see the initial dough. After 2 hours, you'll cover it up completely. You'll have to cover that dough completely. Okay, maybe with a cloth, or maybe you'll put it in an airtight container. All right, and keep it for about 2 hours, and after that you'll observe what happens. You all will see that the dough has risen. All right, so what happens is the yeast ferments the sugar. The yeast ferments the sugar. Okay, it changes, it produces carbon dioxide, due to which the dough rises. All right. Okay, next slide. Okay, all right. So it produces carbon dioxide because of the process of respiration. It produces carbon dioxide, due to which there is a rise in the dough. Next slide. Okay, in the previous class, even I explained about how the yeast produces carbon dioxide. This is the experiment to show how yeast produces carbon dioxide. I'd shown you all 3 test tubes, right? Do you all remember, Janvi and Sakshi? I'd shown you all 3 test tubes. Can you tell me what did I take in the 3 test tubes? Yes, Janvi, say.
I think you took yeast, sugar, water,
Yes.
and a balloon. That was outside the test tube, actually.
Okay. So, so have you all tried that experiment last week? No? Sakshi, you tried it? What happened?
I'm sorry, can you say it again?
Yeah, I asked whether you all tried that experiment last week. Did you all try this experiment?
Yeah, I tried the balloon experiment, it worked.
It worked?
Yes.
Okay. Did not post pictures on the group, so we did not know. Okay. Henceforth, please post pictures so that we can see. Okay.
Okay.
So in one test tube, we had taken sugar solution. Okay, we had taken sugar solution and water. Okay, we did not add maida or atta to it. In the other one, we had taken sugar solution, maida, and water. And we had in one we had added more sugar, and in one we had added less sugar. I'm sorry. We had taken 3 test tubes. One we had not added sugar at all. Okay. In one we had added little bit of sugar, and in the other we had added more of sugar. Okay, and in 2 test tubes we had added yeast. So the one with more sugar had uh, the balloon had inflated more, and one with the less sugar, the balloon had inflated less, because of the carbon dioxide. Okay, carbon dioxide production in the test tube having more sugar was more, Therefore, the inflation of the balloon was more. In the test tube having more sugar. But in the test tube having less sugar, the inflation of the balloon was less. Okay, so that was done in the last class. Anyways, okay, next slide. Okay, so we have seen how carbon dioxide is produced. Due to carbon dioxide, the balloon had inflated. All right. Now um, this is the chemical reaction for fermentation. Okay, how glucose produces ethanol and carbon dioxide. Next slide. All right. Now in this, what you'll have to do, you'll have to. Do you all have any beaker? Yes. Okay. So y'all will take warm water in the beaker. Bring everyone. Bring your beakers.
You can use any container, not necessarily a beaker. Any container
Yeah,
for that
not
matter.
glass container.
A glass container. Nishreya, you have all the supplies?
Yes, ma'am, I do have.
Okay, so get whatever ma'am is asking you to.
Get a glass container. Okay. Yes, Janvi, but you also need warm water. Okay. You'll need a glass container. You'll need warm water. You'll need sugar. These are the three supplies that you'll need. Nishraya, is your water warm? Okay. Okay, let Sakshi get her supplies and then we'll start. So what y'all will do, both of y'all, since y'all have already got your supplies, y'all fill your container 3/4 with water. 3/4 of your container should be filled with water.
The water shouldn't be hot.
Yeah, it should be warm. Okay, don't drink hot water. It y'all use warm water. Sakshi, have you got your container?
Yes, I have got my container, but I forgot the warm water.
Okay. Okay, take some time and go warm water and drink. Fill your container. 3/4 of your container should be filled with warm water. Okay. Nishra, have you done it? Okay. Now what you can do, you can add 2 tablespoons of sugar to it and stir it. 2 tablespoons of sugar and you'll stir it. Stir it well till the sugar completely dissolves in the water. Yeah. Stir it. Keep on stirring it till the sugar completely dissolves in water. When you all are done, tell me that you all are done. Okay, with the stirring of sugar.
I'm done.
Okay. Now what you can do, Janvi, you can add yeast to it.
How much?
About 1.5 tablespoon. Okay. After adding the yeast, you'll stir it again and keep it there for 10 minutes. What you all can do is stir it well, okay? And you all can uh, airtight your container. You all can make your container airtight. How will you all do it? Janvi, what type of a container do you have? Show
I
me.
have a beaker.
You have a beaker. Do you have what is that? Aluminium foil?
Yes, I think so. I'll have to.
So you put aluminium foil and put a rubber band to it, okay, tightly, and keep it aside for about 5 hours. Okay.
Okay.
Okay. Nishraya, show me what type of a container do you have? Okay. Do you have aluminium foil? It's a wide mouthed container. Do you have aluminium foil?
Yes, I do.
Okay. You also do the same thing then. Okay. Yeah. Okay. Sakshi, have you done the okay, you brought warm water now. Is it very hot? No, it must be warm. So you're touching it. Okay. So what you need to do, you take your container, okay, fill it 3/4 with water, with the warm water, okay, and add about 2 tablespoons of sugar to it and stir it well. Okay. Yes. And Janvi, you can secure it with a rubber band tightly. Do you have a rubber band? Okay. Nisha, you also secure it with the rubber band, okay? Sakshi, did you stir the sugar well?
Yes.
Okay. Is the sugar completely dissolved now?
Yes.
Okay. Now you can add yeast to it, about 1.5 tablespoon of yeast to that solution. You add 1.5 tablespoon of yeast and mix it well. Okay.
Okay.
Done.
Done. Okay, you'll have to set it aside for about 4 to 5 hours. After 4 to 5 hours, you'll open it, and you'll will get a very strange smell from it. Okay, that is because the sugar has been converted to alcohol. Okay, the sugar has been converted into alcohol by the yeast. So that is why. So what are we actually doing? We are actually forming alcohol with the help of yeast. Okay, the sugar is getting converted to alcohol. After 4 to 5 hours, you'll open it. Okay, and you'll will get a very strange smell that will be of alcohol, because the yeast will have converted the sugar into alcohol. All right.
Sorry.
Okay, so this is something which what it will look like. I cannot. You all will get the smell yourself. I cannot tell you all about the smell, but yes, it will look something like this. Okay, you all can smell it after 4 to 5 hours. How does it smell? Okay, Sakshi, have you done it?
Yes, but
Uh,
I don't have foil to cover.
okay, show me the container that you have. Okay, you used this container, so what you can do is
Um, Probably use a plastic cover and secure it with a rubber band.
yeah,
I
that will
don't
work.
have foil nor plastic.
Okay, you don't have a polythene bag? No. Hmm. Okay.
A handkerchief,
Yes.
try that out.
Okay.
Okay. So this is what you will observe after 4 to 5 hours. Okay. So this experiment will show us that yeast converts the sugar into alcohol. All right. Now we'll move on to the next experiment. And one very important thing, why I'm telling you all to cover it with an aluminium foil, or you all have to cover it, because yeast will convert it into alcohol only in anaerobic conditions. Can anyone tell me what is anaerobic condition? What do you mean by anaerobic condition? Have you heard the term before? Yes, Anishai, you have heard it? Okay. So anaerobic condition means in the absence of oxygen, you'll have to cut off the supply of oxygen. Okay, so that is why we have to put it in an airtight container to cut off the supply of oxygen. All right, and only in anaerobic condition the yeast will convert the sugar into alcohol. Okay, and that process will be called fermentation. Okay, next slide. Okay. Now, what you will do? Do you have any containers or pots at home? Yes? No? Uh, See, y'all need. If you all have pots, well and good. If you all have containers, it will serve the purpose. Doesn't okay? You all can take two pots like this. If you all have, if you all don't have, you all see what you all can do is, and if you all do not have pots, you all can take. Do you all have plastic bottles, old ones?
Ma'am, is this okay?
Yeah, but that's too small. What? Okay, it will work. But do you have anything like any pot or any plastic bottle will also serve the purpose? Do you have the oil cans, the plastic ones, old ones which are not in use now?
Mom, I have pots, but they are of two different shapes and sizes.
No worries about that. It will work. Okay, no worries about that. About the size, we are not going to see whether you are only going to put biodegradable waste in one and non-biodegradable waste in the other pot. So it doesn't matter. Any other thing like oil cans, the plastic ones that you'll get if you are having. Okay. So Nishraya, do you are going to use these bottles, right?
Yes.
So what you can do you need these bottles again?
No.
No. What okay, you can cut off the top of the bottle, okay? You can cut off the top of the bottle, okay, both the bottles, and you'll have to put soil in both, okay? And in one container, you all are going to put biodegradable waste. That is your kitchen waste, maybe vegetable peels, fruit peels, okay, onion skin, all these things you all are going to put in one pot. And in the other pot, you are going to put, if your container is very small, what you'll do, you'll must be having old any polythene bags. You all must be having. You'll cut it into small pieces. Okay, you all must be having any old plastic use and throw bottles. The mineral water bottle, if you'll have, then y'all can cut it into small pieces. Okay, and y'all can add it to that pot. Any type of non biodegradable waste. It can be iron nails, it can be this old plastic bottles, or it can be polythene bags. Okay, you can add it to another pot. One pot you are going to add kitchen waste, and the other pot you are going to add these things. Okay, and you'll have to keep it as it is for 1 month. It's a very long process, okay, because it takes a very long time uh, to degrade. Okay, the degradation process is not done in 1 or 2 days. It will take 1 month, so you'll have to keep it somewhere and after 1 month you'll have to dig out that soil and see what has happened to the contents that you all had buried 1 month ago. Okay, that is what you'll have to do. So Janvi, can you collect two containers, any containers? It can be plastic containers too, any old ones which you all are not going to use.
I don't have any.
Okay, you don't have anything. What else can work? I think a polythene bag will also work. Not the thin ones. A plastic polythene bag. Do you have that, Janvi? Plastic polythene bag. Sakshi, what about you? Do you have any containers so that you can carry out this experiment?
I'll see. I'll ask my mom.
Okay.
Ma'am, the contents in the glass container are overflowing already.
Okay. Okay. There's something growing. Oats are growing there. All right. So
So,
means you are.
can you show us? Can you show us that, Nishya?
Yes, ma'am. Yeah.
Her yeast is already working. Can you hold it a little higher? Yes. So the yeast has activated and it has started working. Now don't disturb it. Let it eat to its heart's content.
Mom, will a Ziploc bag work?
What, Sakshi?
Will a
Who asked
Ziploc
me?
bag work?
Okay. No, I don't know. It won't work.
So y'all can look for a container. It can be a pot that you use in your garden or in your gallery. It can be a pot like that because uh, taking care of your polythene bag for 1 month will be quite a difficult thing. So what you can do is, if you have a pot, you can arrange for a pot. You just remember the way ma'am is asking you to do, and you carry out this activity. And uh, today is 11th of August, so we will see the results by 15th of September.
Yeah. Okay. Yeah. Did you all understand how you'll have to do it? You'll have to put soil in both the containers. Okay. And you'll have to bury these things in one pot. You'll have to bury kitchen waste. Okay. All the waste from your kitchen. And in the other container, you'll have to bury plastic or polythene bags, whatever non biodegradable waste you'll get. Okay. And after a month, we'll see what has happened to all these contents. What have you all buried? You'll make a note of what you'll have buried. Okay. Just make a note um, of what you'll have buried in which pot. And then after 1 month, we'll see what has degraded and to what extent it has degraded. All right. All right. Next slide. We'll see that the bio. Anyways, you'll observe and then we will discuss on this. Okay. Okay. Now y'all can since y'all cannot be shown what is seen in the microscope. Okay, we'll discuss about how does it look like. Y'all can see this is called a paramecium. Okay, and found it's found in the water bodies, ponds, lakes, all these water bodies. Paramecium is found. See, when you'll see its shape, it looks like the sole of a shoe or a slipper. Okay, like that it looks. It is shaped like that. And the outer covering of the paramecium is called pellicle. The outer covering of the paramecium is called a pellicle. All right. You can see the tiny hair-like structures on the paramecium. They are called cilia. Now, what does the cilia help the paramecium to do? Can you all guess? What might be the function of the cilia?
Movement.
Yes, movement. Okay. So, very good. So, cilia helps in movement of the paramecium. All right. So, this is what the paramecium actually looks, but what it looks under the microscope, you all can see in the uh, adjacent pictures. 40x magnification. When it is magnified up to 40 times, what it will look like, you all can see these are microorganisms. So, it will just be of that size, and when it is magnified up to 100 times, you all can see how it looks like. under the microscope. That is 100x magnification. Okay. So there are different magnifications under the microscope. All right. But 40x, you all will see it 40 times larger. 40 times larger, it will look like this, and 100 times larger, it will look like this, which you'll see in the pictures. All right. And see how you all can see how the paramecium moves in water. When you all collect a sample of pond water, okay, if you all collect muddy water and you all put it on a slide, you all can see paramecium or amoeba. Okay, these are the two microorganisms that you all can see in pond water, or in muddy water you all can see these organisms. Okay, next slide. All right, now we'll discuss about bacteria. Okay, the picture on the right is that of a bacteria. Now, what are the parts of the bacteria? See, inside the bacteria, you all can see some spiral shaped things. Okay, now what is that? That is the DNA of the bacteria. We also have DNA. The genetic material inside us is also DNA. Okay, in the same way, the bacteria also has DNA. But you all can see, have you all studied about the cell previously? Do you all know what is a cell? You all know what's a cell? Tell me, what is a cell?
Ma'am, it's the smallest human body.
Yes, it's the smallest unit of the human body. Okay, have you all read about the parts of the cell? What is inside the cell? Yes. Have you heard about the term nucleus before? Yes. Okay. So you all can see in a nucleus, when you see a nucleus, inside the nucleus, the DNA is there inside the nucleus. But you all can see the bacteria. There is no covering to the DNA. It is all freely placed. Okay. So we can call these type of cells as prokaryotic organisms. These type of cells or organisms as prokaryotic organisms. Okay. And since it has a single cell, it is also called unicellular. So bacteria always has only one cell. So it is unicellular and it is prokaryotic. Prokaryotic means it's a very simple organism. Okay, it does not have any wall to the genetic material, to the DNA. No wall. It is freely placed inside the cytoplasm of the bacteria. Okay, the DNA is freely placed. So what is this type of nucleus called? It is called a nucleoid. It's called a nucleoid. Only the bacteria, okay, they are prokaryotic and they have a nucleoid. Okay, nucleoid means they don't have a well-defined nucleus. So bacteria, they have a nucleoid. And what are the shapes of the bacteria you can see in the pictures? The first one is cocci. That means they are round shaped, okay, or spherical in shape. So these are the shapes. Now bacteria can be single, okay, or else it can form colonies. It can form colonies. Lot of round shaped bacteria can come together and form colonies. Okay, either it can be single or it can form colonies. See, it is called Diplococcus, Staphylococcus. Tetrad means because it is having four round shaped bacteria, so it is tetrad. Okay, in the same way, different types of they form different colonies, we can say. Okay, either one can be single, also it can be two, it can be three, four, and so many in number. Okay, so that is coccus. Now we come to the rod shaped bacteria. You can see how it looks. It is called bacillus. Have you heard about lactobacillus bacteria? Yes. where do you find the lactobacillus bacteria? Where have you heard? Yes,
Curd.
Janvi. Good. Very good. So, lactobacillus bacteria is found in the curd. Why it is called lactobacillus? Lacto refers to the milk product. Okay. So, lacto from milk and bacillus because of the shape. Okay. So, rod shaped bacteria are called bacillus. Okay. They are called bacillus. Then there are some bacteria which are spiral in shape. You all have seen telephone wires or strings. Okay, they are spiral in shape. So some bacteria also spiral in shape. Some bacteria look like commas. Okay, how we put a comma? That type of bacteria are called vibrio. They are called vibrio. All right. Now you can see the picture of the bacteria. It has something called like a tail. That's called flagella. It's called the flagella. Now, can anyone guess what will be the role of the flagella? What is the role? What is the function of the flagella?
To give direction.
Yes, to give direction. Very good. To give direction for movement. Okay, it is the flagella is used to give direction and to help the bacteria to move. But all bacteria don't have flagella. Only a few of them have flagella. Okay. Now you see, bacteria are the smallest of all the microorganisms. Bacteria, out of all the groups of microorganisms, bacteria is the most simple and the smallest of all the microorganisms. Okay. So you see, when you enlarge it to 100 times, see how small does it look? 100x magnification. You all can see, still it is quite small, right? If you magnify it up to 100 times, it looks so small under the microscope. And if you magnify it up to 400 times, it looks in this way, like the second picture. Okay. If you magnify it up to 400 times, it will look like this. Okay. And now, can you tell me what is the shape of the bacteria that's mentioned in the pictures? That's in these two pictures that are at the bottom. What is the shape of the bacteria in both these cases?
Vibrio or bacilli.
Yeah, bacillus. Okay. Okay, that's bacillus. That's a rod-shaped bacteria. Okay, we can move on to the next slide.
One question just popped up in my mind. So Sakshi and Nishraya can answer this question because they had come with us for the industrial tour to the water treatment plant. So do you remember in the laboratory, we had seen colonies of bacteria that they were growing? Do you remember what was it and how were they growing it?
I don't remember exactly.
So it was a testing laboratory, right? So they had taken water sample and they had used something which was like the aloe vera gel. What is that thing called? You remember? They had told us it was agar agar gel, and they had kept some petri dishes in which they had kept agar gel, and they were growing colonies of bacteria, and they had kept it in the incubator. Do you all remember, or it has vanished from your minds? You remember that?
Yes.
Okay, so that is how, like, ma'am was just telling that some bacterias are present in groups. So just came into my mind. Sorry for interrupting.
No issues. Okay. All right. So that was about bacteria. Now we'll discuss about algae. Now the most prominent feature of algae is that they are green in color. So they look like plants because they are green in color. So what is the pigment that must be present here that makes them green in color? What is the pigment? Can anyone tell me? What makes the plants green in color?
Chlorophyll.
Yes. So the chlorophyll pigment makes the algae also green in color. So since they have chlorophyll, they can also prepare their own food like the plants. Okay. The algae can also prepare their own food like the plants. And they are found in the water bodies. You all must have seen ponds comes. A thin layer of green color, a thin green colored layer on the ponds. Okay. See the picture. You all must have seen it somewhere. It's called a pond scum. You all must have seen a thin green colored layer on the water. Okay, so that is algae. And algae may be unicellular. They may have one cell, or they may be multicellular. They may have more than one cell. Okay. How does the algae look under the microscope? See, if it is magnified up to 100 times, it looks like this. Okay. And it is magnified up to 400 times, it looks like this in the next picture. So algae are not at all harmful for us. It is always useful for us. We have already discussed in the last class that how algae can be useful to us. Have you heard about spirulina capsules and all these things? That is algae. Okay. Some people also they consume seaweeds. Some seaweeds they also eat. Not in India, but out of India they eat. They consume seaweeds. Okay, so that acts as a food. Even the agar-agar gel that ma'am discussed about, that also we get from algae. Okay, we get that from algae. Not the green algae. There are different other colors, colored algae also. We get it from the red algae. Okay, so there are different useful. There are different useful effects of algae. Okay, algae is very, very useful to us. All right, next slide. Now, for this experiment, okay, this is a food preservation method. So you all need 4 containers for this. Do you all have 4 containers? Okay, all of you all can put your videos on. Do you all have an apple? No? Nishaya, do you have an apple? Okay. All right. So you all can do it anytime after the class, okay? Janvi and Sakshi. What you'll need to do is, I also do not have an apple. I'll show it to y'all with a potato. What y'all have to do, okay? 4 containers you'll take. In one, you'll add, put sugar solution. Next container, you'll have to put salt solution. Third container, you'll have to put vinegar. And the fourth container, y'all don't have to put anything. It acts as a control. I'll tell you all what's a control. Okay. First, y'all can note it down. Janvi, Sakshi, y'all can note it down. Y'all can carry it out. Okay. So, Nishraya, what you can do is you have to cut your apple into 4 parts. Okay. In the 4 containers, you put your in one container, you have to put sugar solution in one container. Okay, and you put one part of the apple into that. In the other container, you have to put salt solution. One piece of apple you will put into that. In the third container, you will put vinegar. Okay, and in the fourth container, you will put nothing. It will act as a control. What does a control mean? Means if we will see that without any preservative, what will happen to the apple? So that is the role of the control. Okay, means without adding any preservatives, what will be the condition of the food sample, and what will be the difference in the condition of the food sample in all the three containers. We'll observe whether it has been attacked by any microorganism in these three containers. Okay, that we are going to observe. So that you'll have to carry out. Okay, and you'll will observe very minutely what changes have taken place in your food sample. Is it getting attacked by any microorganism, or is it not allowing any microorganism to attack it? So you all will observe in the three containers what is actually the difference in the food sample. If there is any change in color, you'll have to detect that change in color. If there is any change in the size, y'all will also mention about the change in the size of the piece of apple. Okay, if there's any other thing which has grown in your container, that also y'all will mention. So y'all are supposed to observe what are the differences in your food sample in the three containers. In this, obviously, it's not going to remain for a long time, because
Ma'am,
we are not adding any preservative as such. Yes.
ma'am, could you explain what you're putting in the containers? One is sugar solution,
Yeah.
others?
One is sugar solution, one is salt solution, one is vinegar, and the other one there's nothing. Only your food sample.
Okay. Thank you.
And you all can note the differences, okay, in all the containers. What has happened, and you all can note it down. Sakshi?
Yes.
Yeah. So your video is not on, so I couldn't see you. Anyways, what you'll have to do is you'll have to note the observations. All of y'all need to note down the observations, record your observations, and let us know.
So when you make the observation table, uh, write different properties like smell, color, size, okay, and for how long does the food did not change. For example, it will stay for a longer time in either of these three solutions, but in the remaining two it will start rotting early. So try to observe the changes.
Okay. All right. Now, this is what you'll have to do in this experiment. Now, we will also discuss a further research activity. Before that, I will be asking y'all to find out about two questions. Okay, in the next class, y'all we'll be discussing more on it. So y'all have to find out how microbes are. Your first question is how microorganisms can be used for energy production. Okay, so microorganisms can be useful to us. How can they be used in energy production? How can they produce energy? You all are going to find out about that. You all need to research a bit about that. Okay, and you all know that pollution is a major problem in India. Okay, let that be any pollution: air pollution, water pollution, soil pollution. Kanpur is it is ranking the highest in air pollution because of its industries, shoe industry, leather industry. Because of that, Kanpur is highest in air pollution. Water and air pollution goes to Maharashtra. Okay, so these two, the credit goes to Maharashtra because they are producing most of the pollutants in water and air. Okay, not sorry, water and soil, not air. Air pollution is of means highest one is Kanpur, but water and soil pollution, Maharashtra is ranking the highest in water and soil pollution. So how do you think microbes can help in combating pollution? So that's the second question. How do you think, or you all can research more about how microbes can be used to combat pollution on a general level? Let that be any pollution. Okay, these are two questions. So we'll discuss about the further research activity. One second. Now, one more thing you all can do is you all can find out the names of the preservatives used in some of your packed food items like jams. Okay, what are the preservatives that are used? You all must be having a jam container, a sauce, some sauce. You all must be having any canned food items. Okay, you all find out the preservatives that are being used there and note down the names of the preservatives. Okay, so that's your third question. All right. Now we'll discuss about the further research activity. What you'll have to do is, this is, y'all can make a DIY microscope at home. Okay. What y'all can do is, because since y'all cannot observe the things in the microscope because of the COVID and all. Okay. Um, so what y'all can do is y'all can prepare a DIY microscope at home. What y'all can do is, um, if you all have an old laser light, do you all have an old laser light which is no longer in use? No? Okay. Okay, anytime if you all get an old laser light, you all can try this. Okay, what you all will do is, you all need the lens of the old laser light. The lens of the old laser light. Okay, you all have your phone where in the at the back of your phone the camera is there, the camera of the phone. You all place the lens of the old laser light there, and you all I mean you all can secure it with something. You all can secure it with atta or maida or something. Okay, now this link will show you all how to do it. This is a video. Okay, this is a video that will show you all how to do it. What you'll have to do is, you'll have to take your phone. At the back of your phone there's a camera. You all can take out means an old laser light you all can use. You all can take out the lens of the old laser light, put it in the camera and secure it with maybe atta or maida something. Okay, then what you all will do, you all will use a torch light. You all will put the torch light vertically. On top of the torch light you all will put a white paper. Got it? You all will put the torch on. You all will put a small piece of white paper. Then what you all will do, you all must be having these um, plastic bags. Okay, plastic bags that you'll get vegetables and rations and all. You'll get these plastic bags. So you all can cut a piece of plastic bag in the form of a slide. Okay, you all must have seen a slide, right? How you all can cut it in a rectangular manner. Okay, you all can put it on top of the white paper, and then you all will cut a very thin very small piece of, like in the form of a square. You all will, on top of that plastic paper you all can put a drop of water. water means maybe muddy water. You can put a drop, and then you can put that small piece of plastic on top of that plastic which you all had cut in a rectangular way. It acts as a cover slip, and then you all put this light on, and yours mobile, okay, should be in this way. The lens of the can you all see this? The lens should be somewhere here, okay, and you all place the mobile at a height. At a height you all place it, and the torchlight should be here on top. It will act like a slide, and then you all observe. You all can see when you all put the camera mode on, you all can see the microorganisms. Did you all follow what I tried to explain? Did you all follow it? With your camera, you all can have a magnified image. You all can get the magnified view of the microorganisms that are present inside the water, that are present in the water. Because of the laser light, because the laser light is having convex lens. Okay? And the convex lens magnifies images. So it will magnify the water sample, and it will magnify things that are present inside the water. So that is how you will be able to see the microorganisms in your water sample. Okay, so that is how you can prepare a DIY microscope. All right. So did you all understand everything today?
Yeah.
Okay.
I had a question.
These are yes.
What's the role of the first experiment we did? What's the role of sugar in that?
Okay. What see, the role of sugar is to the sugar will only get converted into carbon dioxide. The sugar will break down. The yeast will act on the sugar. Okay, the yeast is going to act on the sugar itself. So the sugar is actually going to be converted. Can we go to some slides before? That you all might have noted the chemical equation. The equation, if you all have noted, the equation was there in, yeah. See what happens. The glucose will break down to ethanol and carbon dioxide. So glucose is the sugar.
Okay.
So that is okay. So the sugar is breaking down to form ethanol, or that is alcohol and carbon dioxide. So in one experiment we saw the evolution of carbon dioxide, and in the other experiment which you all have kept it for you all will keep it for 4 to 5 hours. There we'll see ethanol production. Okay.
Thank you, ma'am.
So any doubts from today's class? No. And have you all noted down all the questions? Okay. So you'll find out about it. We'll discuss about it in the next class. All these answers. Okay. And you all have to also find out and let me know what you all have found out about it. All these answers. Here. All right. Bye. We'll meet in the next class. Another chapter.
Ma'am,
Yes, Janvi.
can you add the meeting link in the group description of the grade 8 KitKat batch, because it's a little hard to find it.
I will do that. I'll do that.
Okay.
Yeah. So whatever questions that we are giving you all from this week, maintain a notebook and write the title of the class, whatever we did. Like we are doing microorganisms, so write the title and write those questions in that notebook and maintain it along with the experiment sheets that you all have. Okay, so very soon we'll be having a forum wherein we'll be asking you to upload these answers online. All right. Okay. Thank you, everyone. Do send us pictures of all the activities that you're carrying out. Bye-bye.
Okay.
Bye.
Bye.